So let's get started. Chapter 3, we're going to be going over energy, chemical reactions, and cellular respiration. So there's a lot of biochemistry in this chapter, and I'm going to try to simplify it as much as I can for you and tell you what parts to really focus on and try to give you the big picture behind everything as we go along. So energy, chemical reactions, and cellular respiration. So all living organisms require energy. They need energy to power their muscle, to pump blood, to absorb nutrients, to breathe, to make new molecules, and to create cellular ion concentrations. Ultimately, it's glucose that is broken down through metabolic pathways that forms this energy. And the energy that we use in cells is ATP. It's known as the energy currency of cells. There are two classes of energy. Everything's either A, potential energy, which is energy stored or energy of position, or B, kinetic energy, energy of motion. So a potential energy example would be a rock up very high on a cliff. If you were to push the rock off, it would fall, converting its potential energy into kinetic energy. Same thing with a stretched rubber band. At rest, the rubber band has no potential energy, but if you stretch it, the position, the stretching of the band creates potential energy, stores potential energy, I should say. An example of kinetic energy would be a bowling ball rolling down a lane. Both can be converted from one class to the other. Now, an ion gradient demonstrates both potential energy and kinetic energy. Here you have a cell membrane, which is a selectively permeable barrier, right? It lets some molecules in, but not others. And this is the channel through which it lets sodium ions in. On the outside of the cell, there's a higher concentration of sodium ions than on the inside of the cell. So sodium wants to flow down its concentration gradient into the cell. Before it does, you'll notice this increased amount of ions on the outside results in potential energy being stored. Now, when this ion channel opens, that potential energy is converted into kinetic energy as the ions flow through the channel into the cell. And that's how ion gradients convert energy. So let's talk just a little bit about chemical energy. This is the most important form of energy in the human body. And what it is it's a form of potential energy. So chemical energy is the energy stored in a molecule's chemical bonds. It's used for movement, molecule synthesis, and like we saw earlier, establishing concentration gradients. It's present in all chemical bonds. Energy is released when bonds are broken during reactions. So just some molecules that function in chemical energy storage. We'll see triglycerides. These are the long-term energy storage in adipose or fat tissue. Glucose, you see glycogen stores in liver and muscle. Remember, glycogen is the storage form of glucose. And then ATP, you'll see, which is stored in all cells, and it's produced continuously and used immediately. Just some different energy forms here to be aware of that your book defines. Electrical energy, this is the movement of charged particles. So like the example we saw a moment ago, the movement of ions across the plasma membrane is an example of electrical energy. So is, of course, electricity. Mechanical energy, this is exhibited by objects in motion due to applied force. So this example would be a muscle contraction for walking. So when a muscle contracts, it converts the chemical energy stored in ATP into mechanical energy, which generates force and produces movement. So if you hear mechanical energy, I really want you just to think muscle contraction, because that's one of the biggest examples we have in our body. Sound energy, this is molecular compression caused by a vibrating object. So sound waves causing vibration of the eardrum. And then radiant energy, energy in the form of electromagnetic waves with differing wavelengths and frequencies. So, for example, visible light focused on the retina. These are just the examples out of your book. You have here showing potential energy stored in the chemical bonds. So when glycogen breaks down into glucose, that releases energy. And then glucose itself has several bonds that break, that produce energy, release energy. On this side, you have examples of kinetic energy that we just talked about. So radiant energy, right? Light coming in and hitting the retina of the eye. Sound energy, where molecules that are vibrating hit an object. Mechanical energy, and just remember muscle contraction. And electrical energy, we see that in the movement of charged particles. For example, the nerve impulse down a neuron. All right, so the laws of thermodynamics. We're going to talk a little bit about the laws of energy. It's important to know that conversion of energy from one form to another in the human body produces heat and helps to maintain homeostasis. Now, what do I mean by that? Well, for example, here we have chemical energy. Let's say this is in the form of that high energy bond in ATP. And you break that bond and you convert that energy to mechanical energy, so now your muscles can contract. Well, heat is given off during that reaction. Heat is given off when sound is converted to electrical energy, or when radiant energy is converted to electrical energy, or when mechanical energy is converted to sound energy. All of these processes produce heat as a byproduct. And that's how you maintain your body temperature. Very important to know, the first law of thermodynamics states energy cannot be created or destroyed. It can only be converted from one form to another. And we see that over here when we're seeing the forms being able to be converted from one to another. But there's the second law of thermodynamics, which means every time energy is transformed from one form to another, some of that energy is converted to heat, which we saw here. Again, this helps us maintain our body temperature. Metabolism is the sum of all biochemical reactions happening in a living organism. Chemical reactions occur when chemical bonds in existing molecular structures are broken and new bonds are formed to produce a different structure. And this is expressed as a chemical equation, where you have the reactants on the left side of the arrow and the products on the right side of the arrow. So here you have reactants. These are substances present prior to the start of a chemical reaction on the left side of the equation here. And then you have products on the right side, which is substances formed by the reaction. And the arrow indicates the reaction direction. So the arrow is going to always be pointing at the products. In a balanced equation, the number of elements are equal on both sides of the reaction. So in the example here, we have calcium, which has a positive 2 charge, plus chloride, which has a negative 1 charge. So we know that there needs to be, it needs to be balanced. So it's going to be 2 negative charges and 2 positive charges, and that will give us a neutral calcium chloride. All right, there's 3 main ways we classify chemical reactions. 1, changes in chemical structure. 2, changes in chemical energy, or 3, whether the reaction is irreversible or reversible. In other words, can those products go back to being reactants? Let's first talk about changes in chemical structure. So in this category, we're going to talk about 3 specific reactions: decomposition reactions, synthesis reactions, and exchange reactions. All right, decomposition reaction. This is when an initial large molecule is broken down into smaller structures. So here you have AB goes to A plus B. So AB is your large molecule, and it's broken down into smaller structures or molecules. For example, we have the hydrolysis reaction of sucrose, which is a disaccharide, right? It has 2 sugars, into glucose and fructose. Both of these are monosaccharides. So we've broken a big molecule, or a bigger molecule, into 2 smaller molecules, and we've used water during that reaction. Catabolism refers to the sum of all decomposition reactions happening in the body. So this is the breaking down of larger molecules into smaller ones. That's catabolism. When 2 or more structures combine to form a larger structure, that's called a synthesis reaction. So here we have 2 structures, A plus B, and they combine to form AB, a larger structure. So an example of this would be a dehydration synthesis reaction forming a dipeptide. Here we have 2 amino acids. They come together and form a dipeptide, and water is given off. That's where the dehydration comes from, remember? The dipeptide, or a very small protein molecule, is a larger structure than the 2 separate amino acids. And this is known as anabolism, the sum of all synthesis reactions in the body. This is where you build complex molecules from simpler molecules. Very important to know the difference between catabolism and anabolism for the exam. Finally, we'll talk about an exchange reaction. This is the most prevalent chemical reaction in the human body. And this is where groups are exchanged between 2 chemical structures. It has both decomposition and synthesis components. So here you see AB plus C goes to A plus BC. B has been exchanged between A and C. So a good example of this would be production of ATP in muscle tissue. So in muscle tissue, creatine phosphate is present. So is ADP, or adenosine diphosphate. And you'll see here there are just 2 phosphates on this molecule. Well, ADP essentially takes this phosphate from creatine phosphate, and now we're left with creatine plus adenosine triphosphate, or ATP. Phosphate has been exchanged between these groups. Okay, question. What type of organic reaction occurs when water is released as 2 functional groups bond together? A substitution reaction? No. A hydrolysis reaction? No. Remember, a hydrolysis reaction is actually the opposite. It's what happens when a larger molecule is broken down into smaller molecules and water is consumed. We're looking for the one where smaller molecules come together to form a larger molecule and water is released. And that's C, condensation, which is another term for dehydration synthesis. Okay, a special type of exchange reaction that occurs in the body is oxidation reduction reactions. We call these redox reactions for short. This is where instead of an atom or a molecule being exchanged between 2 groups or 2 structures, electrons are exchanged between 2 structures. So in an oxidation reaction, the structure that loses an electron is said to be oxidized, whereas in a reduction reaction, a structure that gains an electron is said to be reduced. And the way that I remember these and keep these separate in my head is I remember that reduced is referring to the charge on the molecule. So let's say you started with a neutral molecule and it gained an electron. Its overall charge would be reduced from neutral to -1. Likewise, if you had an ion with a +1 charge and it gained an electron, its overall charge would be reduced from +1 to neutral. So remember, the structure that gains an electron or electrons is the one being reduced. Oxidation-reduction reactions always occur together, and the electrons may be moved alone or with a hydrogen ion. So if it's moved alone, we denote that as just an E with the negative sign to denote electron. But if it's moved with a hydrogen ion, we denote it as H, right? And this is because one hydrogen ion plus one electron equals a hydrogen atom, symbolized by H. Okay, here's an example of a redox reaction. So here you have NAD plus, this is the oxidized form of nicotinamide adenine dinucleotide. It gains 2 hydrogen atoms, or 2 hydrogen ions plus 2 electrons, from an energy-rich molecule such as glucose, and it becomes reduced to NADH. And you can see the change in the bonds here. So NAD plus gains an electron here, and it also gains a hydrogen atom here. So looking on this side, you can see the hydrogen atom here, and you can see the electron here because this positive charge has disappeared. And it also produces hydrogen ion as a byproduct that is released into the local environment. So remember, NAD plus is the oxidized form, and it receives hydrogen atoms from an energy-rich molecule like glucose. And remember that NADH is the reduced form, and it's carrying those electrons essentially from glucose. When it gets to where it's going, which we'll see where it's going here in a little bit, it releases those electrons and becomes the oxidized version of nicotinamide adenine dinucleotide again. All right, the second way we can classify chemical reactions is through changes in chemical energy. So we have exergonic reactions, which are reactions that release energy. Those would be like your decomposition reactions. And we have endergonic reactions, and those are reactions that require energy in order to start or to go. An example of this would be synthesis reactions. So here you see if energy was at 0, this is where we would be, and then you'll see the energy level drop here from reactants, which have higher energy, to products, which have lower energy. And if the products have less energy than the reactants, remember conservation of energy, or the first law of thermodynamics, says that energy is conserved, so it must be released somehow. Here you have energy at 0, and you have reactants down here, and you have the products up here, so the products have more energy than the reactants. So again, conservation of energy, that would mean we had to put energy into this system to get the products up here. And I want you to be able to interpret graphs like this and understand which one is an exergonic or energy releasing reaction, and which one is an endergonic or energy requiring reaction. Finally, we can talk about chemical reactions in terms of whether they're irreversible or reversible. An irreversible reaction has a net loss of reactants and a net gain in products. So this is where A and B goes to AB, or AB goes to A plus B. But you'll see they can't go backwards. So once you've gone through this step, it's irreversible. There's no putting A plus B back together to get AB. A reversible reaction, on the other hand, does not proceed only to the right. It can go both forwards and backwards. Reactants become products, and products become reactants at an equal rate. And this is known as equilibrium, when there's no net change in concentration of either reactants or products. So here we have our reaction, A plus B goes to AB. But you'll see this backwards arrow means that AB can also go to A plus B. And when A plus B to AB and AB to A plus B are at an equal rate, this system is said to be in equilibrium. Reversible reactions will remain at equilibrium until they're disturbed by something. So if you add more reactants to the reaction, it will drive the equation to the right until equilibrium is reached. And vice versa. If you add more products to the equation, it'll drive the reaction to the left towards more reactants. Okay? And the example given here is carbon dioxide reacting with water to form carbonic acid. So there's a forward reaction, but there's also the reverse reaction where carbonic acid breaks down to form carbon dioxide and water. And if it's at equilibrium, both the forward and the backward reactions are happening at an equal rate. Okay, let's take a look at the ATP cycle. So this is a reversible reaction in which adenosine diphosphate plus inorganic phosphate plus energy, so this is going to be electrons from glucose breakdown, goes to ATP plus water. So you have ADP here, and ADP has three parts to it. It has the nitrogenous base, adenine. It has a 5-carbon sugar, ribose, and it has 2 phosphate groups hanging out. When this is combined with inorganic phosphate and energy, water is given off and ATP is formed. This third phosphate is plugged in right here. This requires energy, so it's an endergonic reaction. The reverse direction, on the other hand, is where ATP with water and the release of energy produces ADP plus inorganic phosphate. You'll see that this is an exergonic reaction because it releases energy. It consumes water, making this an hydrolysis reaction. Now, this is just an overview of the type of reactions we've talked about. So we talked about reactions involving changing chemical structure. Those is going to be your decomposition, your synthesis, and your exchange reaction. We talked about changes in chemical energy. This is exergonic versus endergonic. And then we talked about the direction of reaction, whether it's irreversible or more commonly reversible. And I want you to be really familiar with all of these, be able to identify them through a chemical reaction. If I give you a chemical reaction, be able to tell me if it's a decomposition reaction, a synthesis reaction, an exchange reaction. If I give you a graph that shows reactants and products like this, I want you to be able to tell me which one's inorganic and which one's exergonic and why. And I want you to be able to recognize reversible reactions. And if I ask you more bicarbonate is added to the system, I want you to be able to tell me which direction the reaction is going to go. So if you add more bicarbonate or more hydrogen ions, that's going to push this reaction to the left. Whereas if you add more water and more carbon dioxide, that's going to push this reaction to the right. Okay, so reaction rates and activation energy. The reaction rate is the rate at which a chemical reaction takes place, and this is the amount of product over unit time. Every reaction has an activation energy. And this is the energy that must be put into the reactants before the reaction can proceed. So in other words, it's the energy required to break the bonds in a molecule so that new bonds can be formed. And we especially see this with enzymes because enzymes reduce the activation energy, speeding up the reaction. So here is the activation energy of a reaction when an enzyme is not present. That's the pink line. And you'll see that this is the energy of activation up here at the top. This is the activation energy in the presence of an enzyme. Same reaction, but an enzyme is present. And you'll see it lowers the activation energy by this much. And it makes the reaction easier to proceed. So what are enzymes? Well, first of all, they're proteins, and they're biological catalysts that increase the rate of chemical reactions. They decrease activation energy of cellular reactions, like we just talked about. It's important to know that they only facilitate reactions that would already occur, so they're not end-alls, be-alls, magic little particles. They will only work on reactions that would already occur in the first place. They increase the rate of product formation. And in an uncatalyzed reaction, there's no enzyme present, and in a catalyzed reaction, there is enzyme present. So here you can see activation energy of a reaction. So the overall reaction is exergonic, right? Because here's the reactants, and here are the products. The products have less overall energy than the reactants, so energy must be given off or released. That would make this reaction exergonic. But there is this portion here that you have to overcome, and that's the activation energy required to initiate the reaction. So we see we have to put a little energy into the system before we get energy out of it. Here is this reaction when enzyme is not present. Again, see the high activation energy. And then the blue line shows the same reaction with enzyme present, and it's a much lower activation energy. So the reaction proceeds much quicker. So enzymes are proteins with a very specific shape. It's a unique three-dimensional structure in a protein called an active site. That's where the substrate binds. And this is known as the lock and key model. So on an enzyme, it's a very specific shape, right, of the active site. And it can only bind the substrate. And the substrate fits in like a lock and key. And that essentially turns the enzyme on and allows it to speed up the reaction. When the substrate binds to the enzyme, it's known as an enzyme-substrate complex. This is a temporary form in the reaction. And remember, a specific enzyme only catalyzes one specific reaction. So there's an enzyme for just about every reaction in your body. Here's another view of it, of the specificity of the enzyme shape. So here is the unique shape. Here's the active site where the substrate binds. Here's the substrate. See how its protrusions, its pockets and bulges fit perfectly into the active site of the enzyme. This is what allows only a single substrate to bind to the enzyme, and it helps it catalyze only one specific reaction. So a couple of questions here. The addition of a catalyst to a chemical reaction will have what effect? A, increase the energy required for the reaction to take place? No, we know that enzymes decrease the energy required for the reaction to take place. B, increase the time required for the reaction to take place? No, we know that enzymes decrease the time required for the reaction to take place. Does it increase the rate at which the reaction takes place? Yes, it does. That's what enzymes do, or catalysts. Which of the following is not a characteristic of enzymes? A, they change shape when they bind their substrates. That is true. That's called a conformational change. We'll talk about that in a second. B, they can catalyze reactions in both forward and reverse directions. That's true. C, their activity is sensitive to changes in temperature. That is also true, so it must be D. They are always active on more than one kind of substrate. We know that that's false because of the lock and key model, where the substrate fits just so into an enzyme. Okay, so this graphic is showing the mechanism of action for enzymes in decomposition reactions. So this is giving you a decomposition reaction, right? So what you're going to do is you're going to take a bigger molecule and break it down into smaller molecules. That's what decomposition reactions do. Well, how does that occur in the presence of an enzyme? Let's give lactase as an example. So here's the enzyme lactase. It binds with its substrate lactose, forming an enzyme substrate complex. Now, once the substrate binds to the enzyme undergoes what's called a conformational change, meaning that it slightly changes its shape. And see how that puts stress on the bonds of the substrate? Whereas here the bonds were a little bit straighter, a little stronger, but now we've got extra stress on those bonds. That's called an induced fit, where the protein or the enzyme kind of wraps around even more tightly after the substrate binds. It puts stress on those bonds, and those bonds break. And you have your two smaller structures here now, the monosaccharides that have broken off of lactose. They are released once they have broken that bond, and lactase is free to go bind another lactose molecule. So what I want you to know about the induced fit model: when the enzyme binds substrate and undergoes a conformational change, remember that's a change in the enzyme shape, the substrate is held even more closely to the enzyme. This is an induced fit. This causes stress on the bonds in the substrate, reducing the energy required to break the substrate's bonds, or in other words, reducing the activation energy. This is showing essentially the same thing, but with a synthesis reaction. So make sure you study this, this graphic and understand just the steps in the induced fit model and how it results in the product. A little bit about classification and naming of enzymes. Almost all enzyme names end with the suffix ase. Enzymes are classified according to their job category, and you can see here the different job categories. So, for example, a transferase transfers a functional group, and an example of a transferase would be phosphorylase, which transfers a phosphate group. I'm not going to ask you specific questions. I'm not going to ask you to tell me, is a phosphorylase a hydrolase or a transferase? Don't worry about that. But what you need to know for sure is the suffix ase. You need to know that that names an enzyme. And you just need to be aware that enzymes are classified according to these 6 major functions. Okay, so enzymes and reaction rates. I've put a chemical equation here that shows an enzyme plus a substrate going to an enzyme substrate complex, right? That's that temporary form. Let me see if I can find it here. This temporary form, the enzyme substrate complex, that then undergoes a small conformational change, putting stress on the bonds of the substrate, which then leads to enzyme plus product, right? Enzyme plus products. The rate of this chemical reaction can be increased by either 1, increasing the enzyme concentration, or 2, increasing the substrate concentration, right? So if you increase enzymes here, it's going to push this reaction to the right. If you increase substrate here, it's going to push this reaction to the right. But it's important to know that if you increase substrate, you're only going to get an increase up to a certain point, and that point is known as saturation. Saturation occurs when all enzyme molecules are presently engaged in a reaction and they cannot bind more substrate. Here you can see on the x-axis we have concentration of substrate increasing. On the y-axis we have rate of reaction increasing. So as you add more substrate, the rate of reaction increases up to a certain point. And this point is enzyme saturation. After this point, if you increase the concentration of a substrate, there's no change in the rate of reaction. And that's because all enzyme molecules are already occupied working on other substrate molecules. So when you put in the additional substrate, it doesn't have an effect. The rate of a chemical reaction may also be accelerated by temperature. Enzymes function best at their optimal temperature, which in our bodies is about body temperature. It's about 37 degrees Celsius or 98.6 degrees Fahrenheit. Severe increases in temperature can cause protein denaturation with loss of function. What that means is that with severe increase in temperature, the protein is essentially destroyed, or its shape is permanently changed and damaged. And that makes it not functional anymore because it cannot bind its intended molecule that it's supposed to bind. At pH, increasing pH depends on the enzyme. So most enzymes have an optimal pH of about 6 to 8. So if you go too far outside of that pH window, either too acidic or too basic, you're going to denature your enzyme. And that changes the shape of the enzyme permanently, so it cannot bind its substrate. Here you can see the effect of temperature on enzymes. So when it's cold, the 3D shape of protein is more rigid. But as temperature heats up, the 3D shape of protein becomes a little bit more flexible and it's easier for it to bind its substrate. But too hot, and the protein breaks apart, or the enzyme breaks apart. This is denaturing, and it's a permanent change to the structure of the protein in that it can no longer bind its intended substrate. So it no longer works. This is showing the effects of pH on enzyme structure. So if optimum pH is around 7, once you go too far, so too acidic, the enzyme denatures. That's a permanent change in its shape, right? So it cannot bind its substrate. And if you go too basic, the enzyme also denatures, permanently changing its shape, preventing it from binding its substrate. Okay, questions. Enzymes catalyze biochemical reactions by A, lowering the potential energy of the products, B, separating inhibitors from products, C, forming a complex with the products, or D, lowering the activation energy of the reaction. If I want you to take anything away from this, it's this: enzymes lower the activation energy of the reaction, or D. Which of the following will affect enzymatic activity? A, temperature, yes. B, amount of water, no. C, form of the enzyme, no. D, concentration of fatty acids, no. A, temperature will affect the enzymatic activity. So I actually think this is a bad question because of answer C, which is form of the enzyme. You could argue that if they mean by form, they mean structure, that the structure of the enzyme determines its ability to function. So if the structure of the enzyme is altered, it could alter enzymatic activity. So I will not give you any question that's ambiguous like this, don't worry. But just know that temperature affects enzymatic activity. All right, now a little bit about how enzymes are controlled. Let's talk about competitive inhibitors. So a competitive inhibitor resembles the substrate and binds to the active site of the enzyme. Remember, the active site is that specially shaped area that's intended to only bind substrate. Well, a competitive inhibitor is a molecule that resembles the substrate, and so it can slip in and bind to that active site. And when it does that, it blocks the ability of the substrate to bind at the active site. So also good to note that when a competitive inhibitor binds to an enzyme, it does not induce a conformational change in the enzyme. So it's competing with the substrate. The competitive inhibitor competes with the substrate for this active site. Now, the way to overcome this is you can put more substrate into the system, and then that will reduce the probability that an inhibitor, a competitive inhibitor, binds at the active site, right? That makes sense. If there's more substrate floating around, it's more likely to bind to the enzyme than the competitive inhibitor. But if there's greater inhibitor in the system, then it's more likely that the inhibitor will occupy the site. Very, very important to know that a competitive inhibitor interferes with the active site. Non-competitive inhibitors, on the other hand, do not resemble the substrate, and they bind to a site other than the active site. And this site is called an allosteric site. They induce a conformational change to the enzyme and to the active site, so it no longer fits the substrate. So you can see here, the non-competitive inhibitor is binding at a site separate from the active site. This is called the allosteric site. It induces a conformational change in the enzyme, and it changes the way the active site is shaped. So again, the substrate cannot bind to the enzyme. Also note that non-competitive inhibitors are also called allosteric inhibitors. You might hear that term instead on your exam, so it's important to be aware of both. Non-competitive inhibitors are not influenced by the concentration of substrate. So it doesn't matter how much substrate you put into the system, because the inhibitor is binding at a totally different site and changing the structure of the active site. So more substrate will do nothing. Metabolic pathways. Metabolic pathways occur when there are numerous enzymes required to convert a substrate into an end product. So you can see down here you've got enzyme 1, takes in the first substrate and produces an intermediate substrate A. Intermediate substrate A goes on to another enzyme, gets converted into intermediate substrate B. Substrate B goes to enzyme 3, produces intermediate substrate C, which goes on to the final enzyme and becomes the final product or the end product. Now note here, how are enzymes regulated? How do you shut this pathway off? Well, as the end product builds up, some of it goes back to the first enzyme in the pathway and plugs into an allosteric site, right? That's a site that's different than the active site. When it binds to the active site, when it binds to the allosteric site, it induces a conformational change, and the enzyme changes the shape of that active site, and the initial substrate can no longer bind. This process is negative feedback control. So this is an example of negative feedback, and is an example of the homeostatic mechanism of metabolic pathways. Remember, the end product of most metabolic pathways inhibit that metabolic pathway. Okay, so let's take everything we just learned and apply it to cellular respiration. Cellular respiration is a multi-step metabolic pathway breaking down molecules by a series of enzymes, and its ultimate goal is to produce ATP, or energy, for the cell. The cellular respiration pathways, organic molecules are oxidized, or they give up electrons, and they're disassembled by a series of enzymes. These pathways are exergonic, that is, they release energy, and the energy that's released is used to synthesize ATP, which is an endergonic process. Oxygen is required for cellular respiration, and it occurs inside of the cell. So the overall reaction would be a glucose molecule plus 6 O2 goes to 6 carbon dioxides and 6 waters. Okay, so big picture thing. Cellular respiration is the reason why you need to breathe in oxygen, right? You got to bring oxygen into the system to move the reaction forward. But you've also got to breathe off carbon dioxide because this CO2 builds up in your cells and your tissues. This graphic is just an overview of the cellular respiration pathways. So the first step, you have glycolysis. This is an ancient pathway. It can proceed in the presence of oxygen or in the absence of oxygen. It occurs in the cytosol. That's the liquid part of the cell. Let's see, it's the breakdown of glucose. Next, you go to an intermediate stage, then to the citric acid cycle, where carbon dioxide is produced. ATP is produced in both glycolysis, the citric acid cycle as well. And then Electron carriers are also produced in glycolysis and citric acid cycle, and they take their electrons all the way to the electron transport system, or the electron transport chain, where they donate their electrons to oxygen and in the process make ATP. Glycolysis, the first step. Glucose is broken down to produce energy, so this is exergonic. Does not require oxygen, so is an anaerobic process. It occurs in the cytosol. This is important to know. There will be questions asking you where these metabolic pathways occur. There are 10 different enzymes required for glycolysis, and the overall reaction is one glucose molecule goes to two pyruvate molecules, two net ATP, and two NADH molecules. All right, so let's look at glycolysis. Okay, so let's talk about the first 5 steps of glycolysis. Ultimately, what happens here is glucose is split into 2 molecules of glyceraldehyde 3-phosphate, and 2 ATP are invested, one at step 1 and the other one at step 3. The reason this happens is the phosphate groups are added to break down the products of glucose, and we'll see this in just a second. So look here, glucose has 6 carbons. ATP is added, now we have glucose 6-phosphate. It undergoes a reaction with an enzyme and changes to a fructose 6-phosphate. Same number of carbons, just a different structure. And here in step 3, here comes ATP again. So instead of here we have just 1 phosphate, now we have 2 phosphate groups. So we have this symmetric fructose molecule. Aldolase comes in and it splits this 6-carbon molecule into 2 3-carbon molecules here. And you'll see dihydroxyacetone phosphate isomerizes to glyceraldehyde 3-phosphate, and now you've got 2 G3P molecules. Steps 6 through 7, this occurs twice in glucose oxidation because that fructose molecule was split into 2 molecules of G3P, right? So it goes from 1 molecule to now 2 molecules. So steps 6 through 7 will occur twice. So in step 6, an unattached inorganic phosphate, that's this right here, is added to the substrate. So we had glyceraldehyde 3-phosphate with 1 phosphate, and now see it's got 2 phosphates. 2 hydrogen atoms, so 2 electrons and 2 hydrogen ions, are released to NAD+. So here's NAD+. It gains those electrons and 1 hydrogen ion. And remember that other hydrogen ion is given off to the local environment from this reaction here. Okay, so gains an electron and a hydrogen ion here, and then you're left with NADH plus a hydrogen ion. So that reaction is happening at this step here, where NAD, the oxidized form of NAD, is reduced. NADH, what it does is it ends up in the electron transport chain and donates its electrons to oxygen, and we'll talk about that here in just a little bit. In step 7, an inorganic phosphate is transferred to ADP. So to form ATP. So here's ADP turning into ATP. So now we've got, remember this happens twice, so 2 ATP molecules have now been formed. That makes up the ATP that we invested in steps 1 and 3. Glycolysis, steps 8 through 10, remember, occurs twice in glucose oxidation. Step 8, we have some isomerization. Step 9, there's a loss of a water molecule. And step 10, inorganic phosphate is transferred to ADP to form ATP here. So here's our net gain of 2 ATP. That's where it comes from step 10. The fate of pyruvate. So what you end up with at the very end of glycolysis is 2 pyruvate molecules. And pyruvate can go on to do a couple of different things depending on if oxygen is available. So if oxygen is available, pyruvate goes to the citric acid cycle. If there's no oxygen available, pyruvate goes to the lactic acid cycle. So I need you to know this for sure. So in summary, glycolysis is a metabolic process. It occurs in the cytosol or the cytoplasm, and it does not require oxygen. Substrates involved are glucose, right? Because we're breaking down glucose and getting energy out of it. And 2-NAD, this gets reduced to NADH. So our final products are those 2 pyruvate molecules, net 2 ATP formed, right? We had 2 invested and 4 formed, and we had 2 NADH. And this is an electron carrier that's going to go to the electron transport chain and give its electrons off, and in the process of doing that, ATP will be synthesized. Remember that pyruvate is the final product of glycolysis. And then where pyruvate goes depends on if oxygen is available. So if there's oxygen, pyruvate goes to the mitochondria and undergoes a citric acid cycle. But if there's not enough oxygen available, pyruvate goes to the lactic acid cycle and is converted to lactate. The lactic acid cycle doesn't result in ATP production, but it does regenerate NAD, and that's for its use again in glycolysis. We recycle many of these molecules because the body does not like to waste. Here are the remaining stages of cellular respiration. We have the intermediate stage, the citric acid cycle, and the electron transport system. All of these stages are aerobic and all occur within the mitochondria. So let's just briefly discuss the anatomy of a mitochondrion. So it's a double membrane structure. It's composed of an inner membrane, that's this thing here, and an outer membrane, this outer part here. The inner folds are called cristae. Then there's this space in between the inner membrane and the outer membrane. It's called the outer compartment, or this diagram calls it the outer chamber. It's also called the intermembrane space. Then on the innermost part, inside the inner membrane, is the mitochondrial matrix. And you'll notice oxidative phosphorylation enzymes, we're about to talk about oxidative phosphorylation, they are embedded in the inner membrane. Okay, the intermediate stage. This is the link between glycolysis and the citric acid cycle. It occurs in the mitochondria, and the substrates are pyruvate, right? Those 2 pyruvate from glycolysis, and coenzyme A and NAD. The final products are acetyl-CoA, which has 2 carbons, NADH, right, an electron carrier that goes onto the electron transport chain, and carbon dioxide, a waste product. And you can see pyruvate, 3 carbons. With the use of pyruvate dehydrogenase, goes to a 2-carbon structure plus the 1 carbon dioxide. So there's your 3 carbons. And then the electrons have been shuttled to NADH. After that, acetyl-CoA goes on to join the cyclic metabolic pathway, the citric acid cycle. There are 9 enzymes in the mitochondrial matrix that make up the citric acid cycle. And in the citric acid cycle, acetyl-CoA, a 2-carbon molecule, overall gets converted to 2 CO2 molecules, so waste products here. CoA is released, coenzyme A, so it can go on to complete the citric acid cycle again. And you produce a little bit of ATP, 1 ATP. You produce a lot of NADH, and you produce a little bit of this, FADH2, which is very similar to NADH in that it transports electrons to the electron transport chain. Oxaloacetate, that's generated at the end of the cycle. It's a 4-carbon structure, and then it continues in the cycle to join with acetyl-CoA to make a 6-carbon structure. The 6-carbon structure loses a carbon in the form of carbon dioxide. Now it's a 5-carbon structure. Then it loses another carbon in the form of carbon dioxide. Now it's a 4-carbon structure. And it's a 4-carbon structure for this whole part of the citric acid cycle until it's essentially restructured into OAA. And then OAA continues the cycle again. So where does acetyl-CoA in the citric acid cycle come from? Well, it comes from the intermediate stage from pyruvate, which we just saw. But it can also be a result of conversion of fatty acids and or some amino acids into acetyl-CoA. So the conversion of fatty acids into acetyl-CoA is known as beta oxidation. You're essentially breaking off small pieces of the fatty acid, and you're creating an acetyl-CoA molecule that can then go on to the citric acid cycle to produce energy. So that's how breaking down fat helps produce energy. So in summary, the citric acid cycle occurs in the mitochondrial matrix. It requires oxygen. The substrate is acetyl-CoA. And the products per each pyruvate, right, and there's 2 pyruvate per glucose molecule. The products per 1 pyruvate are 2 carbon dioxides, that's the waste products, and 1 coenzyme A, and that's goes on to be recycled. And then you've got energy. You've got 1 ATP that's formed. You've got 3 NADH and 1 FADH2. Both of those will take electrons to the electron transport chain. You've got oxaloacetate involved in the first step and regenerated in the last step. And you've got 2 turns for every 1 glucose molecule, and this is because 2 pyruvate were produced in glycolysis. So the overall energy produced is 2 ATP, 6 NADH, 2 FADH2. What I really want you to take away from this is that a little bit of ATP is produced in the citric acid cycle, but it's mostly these electron-rich molecules that are going to go on to the electron transport chain. Okay, so what do I want you to know from this chart? Well, the first 4 items here, if you know these really well and you understand where they come from, you're going to do well on questions over this section. You need to know these first 2 for sure, these first 2 rows. And then I would definitely know that ATP is made, but understand that the other molecules are also produced and they go onto the electron transport chain, the NADH and the FADH2. They are made, they carry electrons to the electron transport chain, where energy, or ATP, is synthesized. And then I would know that glycolysis can be aerobic or anaerobic, but the intermediate stage in the citric acid cycle must be aerobic. So what exactly is the function of the electron transport chain? Well, it's to transfer electrons from NADH and FADH2. The energy released from this electron transfer is used to make ATP. The enzymes for the electron transport chain are located in the inner folded membrane of the mitochondria in the cristae. You'll see hydrogen pumps, that's these channels here. You'll see electron carriers, that's symbolized here by Q and C. And you'll see ATP synthetase enzymes, which here's one such enzyme. The series of hydrogen pumps and electron carriers is called the electron transport chain. So the number of ATP molecules that are generated in the chain depends on the entry point of electrons into the chain. So electrons from NADH enter at the first pump, right here, this first hydrogen pump, and it goes through 3 whole hydrogen pumps. That generates 3 ATP molecules, whereas electrons from FADH2 enter at the second pump, so it only generates 2 ATP molecules. So NADH is slightly more energetic than FADH2 as far as the ability to transport electrons and create ATP. This is a different diagram of the electron transport chain. So let's go over it real quickly. You've got your enzymes involved in the process. So you've got complex 1, complex 2, complex 3, complex 4. You have electron shuttles. It's coenzyme Q and cytochrome C. And you have ATP synthase. This is the one that, this is the enzyme that makes ATP. So what happens is you've got NADH, right, the reduced form of NAD, comes in, interacts with enzyme 1, and that induces a conformational change in the enzyme, and it opens up a channel. And the channel allows hydrogen ions through. And you'll see that with FADH2 as well. When we get to here, the electrons are shuttled to coenzyme Q and then shuttled to complex 3. Complex 3 undergoes a conformational change, opens up its channel, and allows hydrogen ions to flow through. Okay? Same thing with complex 4. When electrons arrive at complex 4 from cytochrome C, they induce a conformational change and hydrogen ions are pumped through. Now, why are the hydrogen ions being pumped through like this? Well, let's take a look. This is the inner mitochondrial membrane. This side here is the innermost compartment of the mitochondria, or the mitochondrial matrix. And this is the intermembrane space. And what happens is you're creating a hydrogen ion gradient. You're putting more hydrogen ions in the intermembrane space than are on the matrix side. So these hydrogen ions want to flow down their concentration gradient into the mitochondrial matrix. And when they do that, they produce ATP through ATP synthase. So the hydrogen ions essentially are the kinetic energy, their kinetic energy flowing through here, power ATP synthase, and allow ADP and inorganic phosphate to become ATP. You'll note here at complex 4 that 2 hydrogen ions plus a half O2 goes to H2O. What you need to know about this is that these electrons that have been shuttled in and shuttled across these complexes eventually go to oxygen, and water is produced. Oxygen is the final electron acceptor in the electron transport chain. Okay, well, what is oxidative phosphorylation? Well, as the name implies, it involves oxidation. So it occurs during electron transport system, and oxygen is the final electron acceptor in a series of redox reactions. So like we just saw, where the electrons are shuttled through the electron transport chain and given to oxygen, and ATP is formed. You, on the other hand, can have substrate level phosphorylation, and this forms ATP from energy directly released from a substrate, and we saw this in the steps in glycolysis and in the citric acid cycle. ATP production, so ATP in the glucose breakdown. This is sort of the sum of all the ATP that gets made. So substrate level phosphorylation, 2 ATP are formed in glycolysis, and 2 ATP are formed in the citric acid cycle, right? 1 ATP per turn of the citric acid cycle, and there's 2 turns. Why? Because there are 2 pyruvate molecules formed at the end of glycolysis. Oxidative phosphorylation, on the other hand, makes a lot of ATP, and this is where those electron carriers come in and donate their electrons to the complexes in the electron transport chain, and eventually those electrons go on to be donated to oxygen. So the total is 4 ATP plus 34 ATP. That's a total of 38 ATP that have been produced. However, there's about a net of 30 ATP per 1 glucose molecule, and this is because some ATP is used to bring pyruvate and NADH into the mitochondrion from the cytosol. Just a clinical note here, cyanide poisoning, it's lethal. Symptoms include excessive sleepiness, rapid heart rate, difficulty breathing, headaches, and seizures. And why this occurs is because it blocks the transfer of electrons from complex 4 to oxygen. So it blocks this step right here is where cyanide halts this process. And if cyanide blocks this step's not going to move forward, and this step's not going to move forward, and this step's not going to move forward. And it basically shuts down your entire electron transport chain almost instantaneously. So the effects are the same as if oxygen were completely removed. This diagram just shows the summary of the stages of cellular respiration and where they're located. So glycolysis, it's showing here, is out in the cytosol, and it produces 2-pyruvate. Pyruvate goes on to the mitochondrion, where it undergoes the intermediate stage, where pyruvate gets converted to acetyl-CoA. And then acetyl-CoA enters the citric acid cycle, produces a little bit of ATP and a lot of the electron carriers. And then showing here the electrons coming in to the complexes of the electron transport chain and producing ATP. And note that this is on the inner mitochondrial membrane, and the intermediate stage and the citric acid cycle are both in the matrix. Okay, we mentioned before the fate of pyruvate when there's insufficient oxygen, but we'll just kind of recap here. So in an environment of insufficient oxygen, the activity of the electron transport chain decreases, right? So levels of NADH and FADH2 will accumulate, and there will be decreased levels of NAD and FAD, the oxidized versions. So the cell will become more dependent upon glycolysis because glycolysis is the only step that can continue in insufficient oxygen. And glycolysis requires NAD to continue. So if left untouched, this glycolysis would eventually shut down due to lack of NAD. So NAD must be regenerated for glycolysis to continue. So we go to the lactic acid cycle. This is where pyruvate is converted to lactate. NADH is oxidized to NAD, so it loses electrons, right, and becomes oxidized. This enables glycolysis to continue because now you have more NAD that can be used. Now remember, glycolysis is not as efficient as oxidative phosphorylation. Only 2 net ATP are generated versus 30 net ATP with oxygen. So this will impact individuals who have a decreased ability to deliver oxygen to the cells. So in a clinical setting, you might see a person who is unable to deliver oxygen to the cells because of shock. Perhaps they're hypovolemic, so they don't have enough blood to perfuse or give oxygen to their tissues and their cells. So one thing you might do in a patient who is in shock is you might check their serum lactic acid, or how much lactic acid has built up in the bloodstream. And an increase in lactic acid in the bloodstream would suggest that these patients are hypoperfusing. In other words, they're not getting oxygen to the electron transport chain, so they're having to undergo glycolysis, anaerobic glycolysis. And you can see down here pyruvate with lactate dehydrogenase, and NADH goes to lactate and NAD+.